So when Al asked me to give this plenary, he said that he did want me to discuss this kind of emerging phenomenon around the country, that these Pseudo blooms do seem to be expanding. And so because my wheelhouse for many years has been in the prediction realm, I'm going to do this in the context of prediction. So I'm going to try to talk a bit about the ecology, but it's not going to be a survey of everything that we know about Pseudo. But I'll try to put that, pepper the ecology throughout the talk as we talk about the ways that we could predict these blooms, and maybe provide a framework for thinking about that prediction. So here along the bottom, just thought it would be nice to illustrate how far we've come. Really been an explosion of Pseudo research in the last 20 years. Go back to the 19th century, we've got these beautiful idealized drawings. It kind of feels like there are new, in every field of HABs, it feels like there's a new species a minute. There's a brand new species of Pseudo identified recently. And just, gorgeous imagery. It's probably the last time you're going to see Pseudo pictures in this talk. So wrap your mind about what they look like. Big diatom, long chains. So I was being pretty flip and cute in my abstract when I decided to misappropriate the term pseudoscience for the science of Pseudo. In no way am I trying to denigrate what we do. But as I was poking around the internet, I found this pretty funny article in Gizmodo. And thought, I'm misappropriating this term, but this kind of fits. It's kind of funny. These blooms, we used to think of Pseudo blooms as these really productive, powerful, amazing things, because for a long time they were just part of the diatom community, say in California along the West Coast. But sometimes they do get mean and ugly and aggressive, and they do have to be put down. Do we put them down? No. This is not a mitigation talk. This is really a talk about strategies for understanding when and how and why they get mean and aggressive. I think the more funny thing about that article was the top 10 list of things that this person called out as the big pseudoscience theories of our time. They were pretty hilarious, but even more interesting was that the number one was toxins. So I thought, well, that's interesting. Let's read a little bit more about toxins. And these are the definitions that she put forth to differentiate a valid definition of a toxin from a pseudoscience definition of a toxin. And again, I lived in Santa Cruz a long time. I'm well acquainted with the bottom one. And so I'm thinking about this. I thought, okay, maybe this analogy is breaking down now. Probably not going to use the term evil pixie dust. But then I thought about the fact that what is the struggle for pseudoscience? For us, it really is that we don't know exactly how, when, and why domoic acid is produced. And this is the struggle, because we do want to predict it. We want to create tools that are useful for managers, that are useful for science, and we're doing it sort of semi-blind. But I'll go about this with the pragmatic approach that it may still be possible. So what do we know about Pseudo? There's a lot, and this is not going to be an exhaustive list, but I pulled the most recent map from the National Office for HABs to just give you a geographic sense of, at least from the point of view of when domoic acid reaches shellfish, what those events have looked like. And this map is a nice illustration of that through 2015. And I'm going to go around the country, and we're just going to talk a little bit about the ecology and what we think we know. If I miss anything, please shout. If I offend you because I didn't include your research, please shout. I really didn't think about this in a truly exhaustive way, but I thought about the things that came to my mind first. When it comes to Washington State, there's clearly a role for retention, a role for eddies, the Juan de Fuca eddy. Nutrients, it's been a little harder to pin down. I think there's been some association in the literature with silicic acid to nitrate. It's been tricky to see that, I think, on an interannual basis. Again, along the Pacific Northwest coast, oh yeah, this is another one of those Mac PC things. Along the Pacific Northwest coast, what you would see here is that onshore flow and river flow are also relevant. So as you're at the border to Washington, Oregon, the Columbia River is very important for determining what those plumes are doing to the water. If you've got the straight coastline and not a lot of retention along Oregon coastline, those downwelling events and the onshore flow are crucial. Maureen McKibben put out a beautiful paper in PNAS about temperature, so looking at these low-frequency warming events, El Nino, and how that is associated with this longer-term variability of when the shellfish are toxic in Oregon. Moving down into California, I would say we have put a little more emphasis on nutrients in our studies over the years there. We have a lot of nice long time series that get at nutrients. We've seen some sort of relationship with reduced silicic acid. We've seen a relationship with regenerated nitrogen. A lot of that work comes from Meredith Howard. Some links to iron. I think that's been a little more in the lab than in the field, but as we know, there's a nice body of literature connecting domoic acid production to iron. As far as temperature goes, there are a lot of long time series that look at the upwelling signature of Pseudo. So we do see, as that temperature drops and maybe starts to warm a little bit into the post-upwelling phase, we see Pseudo blooms. But then, as you all know, or hopefully know, and I'll show you again later from Ryan McCabe's recent paper, there is also a strong connection in 2015 to warming and the Pseudo bloom. So I think temperature is a little bit confusing, and it does co-vary with a lot of things. We also have retention. Our hot spots, for the most part throughout the West Coast, are tied to things like embayments and eddies. Moving into Alaska, I think that despite the fact that you see quite a few dots here or bubbles for ASP events, we don't know a lot about Pseudo in Alaska. Esther Kennedy gave a great talk about the HAB work that's being done there, and I think that there will be an explosion of knowledge in the near future. Moving into the Gulf of Mexico, where I think the first two closures were actually in 2013 and 2014, there has been a long history of studying Pseudo in Alabama and then a bit in Florida. I think of Justin Leifer's papers on groundwater discharge that were very thought-provoking, and then we go back to some of the seminal work of Quay Dortch and Mike Parsons looking at trends in Pseudo with respect to runoff from the Gulf and from the Mississippi River into the Gulf and all of the ensuing impacts that has. Now, some of these events are not represented on this map. We now have Rhode Island this year and last year. Chesapeake Bay is an interesting one because there is a long time series of Pseudo there, even low levels of domoic acid, but no real ASP events or shellfish in the toxins. Ann Thesson's work points to kind of some possibility that nitrogen matters, and I'll loop back around to the Chesapeake Bay a little bit later. But the idea there is that we may be seeing ASP events there as things start to change in the mid-Atlantic. And then I'm going to end here with the Gulf of Maine, which is the newest big deal. There were a lot of events. You can see that ASP has been around. We know that Pseudo has been around. But something has changed in the fact that we now have closures there. So we'll get back into that, potentially circulation, potentially nutrient shifts. But we do know that things are getting worse. At least that's what this map would indicate. As we go around the country and we think about the emergence of these things, it seems like things are getting worse. And we do have some data to back that up, at least in California. So I'm going to go back to a project that I was heavily involved in some years ago, where we examined what that historical record of Pseudo events and domoic acid production look like in sediment traps in the Santa Barbara Basin. And so that yellow triangle is the sediment trap that is pretty much right under the middle of a long-running time series called Plumes and Blooms, which is the red dots are indicating, at least the ones in the middle of the channel. That's Dave Siegel's program. And that sediment trap, we had two of them at 150 meters and another at 540 meters. And there was a nice archive going back to a lot of the early work that Bob Fennell had done, and when Claudia Benitez Nelson came on the scene and helped take over that project, there was an opportunity to go back and pull out all those poisoned samples and see if it's possible to even extract the domoic acid signal. And this was done, as the last slide indicated, with a lot of help from Steve Morton, who did a lot of the, I would say, the imagery and image analysis for this project. As we realized we could get the Pseudo out of these traps, as well as out of the sediments, not just the traps, you can see that there's something going on here. It's pretty obvious that there's not a lot going on before 2000, but there's a lot happening after 2000. And this time series only goes to 2010. We've continued to do this, and the trend is the same, that right around 2000, these things became annual occurrences. And I think that this story has been repeated already in this meeting several times, that we thought of these things as fairly rare, maybe 3 to 5 year events prior to that time. We only really started seeing them in California in 1991, and that was the first time we saw it outside of, say, the Canada region when we saw it, Prince Edward Islands in the late 80s. So the point I'm trying to make here is that this is not maybe just an artifact of the sediment trap time series where you can see in the black bars that we had a lot of gaps. I mean, it's not as if we have a perfect record. But the sediment core work that's been done by other investigators is very consistent with this pattern. And it looks at a 1,000-year time series in the sediments, and you can see that around 2000 in that time record, things really do blow up with respect to Pseudo in the Santa Barbara Basin. So there's some sort of an interesting trend here. And the domoic acid in those sediment traps is also pretty revelatory, because prior to that 2000 kind of step change, there is domoic acid present, and we know that we had some pretty toxic blooms. For instance, the 98 bloom, that was quite significant. But then those toxin levels just jump up an order of magnitude. So we see things are getting more toxic. They're not just more frequent, they seem to be more toxic. And I can say this with confidence, because now that we're measuring these blooms even more, and particularly post-2015, we're seeing some really incredible levels of domoic acid. When we started this work in the early 2000s, thinking about what makes an important bloom or when are we going to see ecosystem impacts, we started thinking about levels on the order of 500 nanograms per liter. We thought, okay, this is probably a good place, or maybe 1,000, to think about ecosystem impacts. And now we're measuring things on the order of 100,000 nanograms per liter in some of these hotspots. It's possible we're just getting better and measuring more, but it also really does seem to hold up that things are getting worse. Claudia and I had the chance to then collaborate a little bit with Stephen Bograd at Fisheries, and he put together some pretty nice analysis of Cal Coffee region, those data sets, and really looking at the surface water with respect to the undercurrent water, which the California undercurrent is the source of upwelling. Around 100 to 200 meters is the sort of central bolus of the California undercurrent. That is where we see the dominant input of nutrients to support these blooms along the California coast anyway. And I'm just illustrating here some of the changes that have happened over this 1984 to 2012 time period. That sigma 26.5, that's that isopycnal that corresponds pretty well with the center of the California undercurrent. And one of the things that jumped out at us in particular was this dramatic change in the levels of silicic acid to nitrate at that depth. That there have been these pretty dramatic changes. They are sort of secular in nature. This doesn't indicate that there was some step change in 2000 that might drive the pattern I showed you in the previous slides, but it does suggest that something is changing. And this from all the work that's been done, this seems to connect back to what are the source waters to those source waters, which is the equator. And this suggests that there are some global biogeochemical changes happening, or at least basin scale biogeochemical changes happening that may be tied into some of the changes that we are seeing along the entire West Coast. So what happened in 2015? I'll just briefly go into this for all of you who have not seen this great paper that does a nice job going through the available data sets and describing how things got blobular. We all heard about the warm blob, the Pacific warm anomaly, that took over the North Pacific and spread along the entire West Coast and was completely coincident with the development of the most unprecedented Pseudo bloom we've seen in the region that spread from Santa Barbara to Alaska almost simultaneously. You can see the warming temperatures in the NDBC buoys, and then, of course, widespread ecosystem impacts that were well documented in this paper throughout the entire food web. And, of course, the Dungeness crab closure. This was West Coast wide, but in California, we had at least $60 million losses just in California from that closure, which lasted the entire season and then trickled into other times in 2016. So there have been about 2 years of really hard hits for the Dungeness crab industry, as well as the rock crabs. So I've already mentioned that temperature was involved. It's pretty clear from the association of just the coincidence of the event with the warming that there's something going on there. So I think this was already mentioned when we were discussing the Quay and with Mark Settleson that the event response funds were really crucial to getting out there and documenting this bloom when it was happening. So Northwest Fisheries Science Center was able to piggyback on an already existing fisheries cruise on the Shimada, the RV Shimada, and you're seeing the results of the Pseudo survey along the cruise. Of course, it took a long time to do it. doing what everybody else did, June. By the time you get up here, the species assemblages have changed. We do know that in May, when this bloom started, there was a lot of P. australis everywhere. So you can imagine that over the course of the bloom, and as those nutrient dynamics are changing and temperatures changing, that things start to change. We don't get a perfect snapshot of what was happening when all of the toxin production was really at its peak. But you can see that there is something going on here, potentially with temperature. Using growth temperature curves, this group was able to point out that if you just look at these isolates in a lab with respect to temperature, there is an optimum. We've talked about this a lot. Fairly close to that 15-degree max that Ted Smeda emphasized, maybe a little bit higher. But if you were to then take this and kind of create an index of potential growth that's just reliant on that temperature, you can make a pretty nice matchup with the ENSO signature as well. So maybe there is something there along the lines of what Morgane showed in her paper in Oregon. And then that might explain the ability of this bloom to go so crazy with higher temperatures. But it also is an interesting question because a hallmark of this bloom is that the absence of any kind of Pseudo Nitzschia bloom in Southern California. So south of Santa Barbara Channel, where waters were clearly warm, we didn't see anything. And so one of the big questions there would be, was it too warm to support Pseudo Nitzschia growth? And nutrients are also important. You can see the excess nutrient experiments do lead to increased DA production. So thinking along the lines of nutrients, John Ryan then explored this a little further and saw that in that bloom, in fact, in Monterey Bay, particularly looking at things like the M1 time series and other platforms that they had out at the time, that upwelling was actually pretty moderate. We didn't have a lot of suppressed upwelling. Waters were relatively cool, particularly in May when the bloom started. And temperatures really didn't reach the optimal region of the growth rate curve until summer. So warming peaked in September, and that was as that bloom was subsiding. And so he then went back and had the benefit of this long-running nutrient time series from the M1 mooring in Monterey Bay run by Mbari, and was able to show that there have been some pretty interesting anomalies, particularly for nitrate, 92-93, El Nino, 97-98, El Nino, and then 2014-2015, the Pacific Warm anomaly. And you can see those big reductions in nitrate. What you also see are reductions in silicic acid, particularly in 2014-2015, which was when we saw the biggest anomalies, both in silicic acid and then the ratio to nitrate. So he draws the conclusion that maybe that was an important driver for this bloom, that at least in May when the bloom was starting and temperatures weren't so warm relative to background in that sort of narrow strip of upwelling along the coast, that maybe these nutrients played a role. But as the bloom went on, they like the warming, they grow well, and you keep getting these pulses of nutrients with upwelling. The thermocline was actually fairly suppressed, and that's why you are seeing this sort of low nutrient regime that keeps coming up. So maybe that was actually allowing Pseudo Nitzschia to hang on, be competitive at low silicic acid levels. So that's just what I said. So given this complexity, we have to think about our model that we've been working on, which is, were we able to predict the DA spatial variability in 2015? Sorry that some of my titles are cut off here. A hallmark of this bloom, too, was this late spring transition. Usually we see this maybe more in April, we'd see this bloom, or late March, but it started in May. We capture that with the California Harmful Algae Risk Mapping System. I'm showing you a mean for the spring season on the left and then the May mean on the right. And we really didn't see a whole lot going on until May in the model. So it's heartening to see this kind of seasonal variability in phenology picked up. We then matched this with that cruise of opportunity that I mentioned. All those domoic acid samples from the RV Shimada gave us a nice opportunity to do an offshore validation, which we rarely have the opportunity to do. And so what you're seeing here are those matchups pulled out for each time space point on the left for the model. Accuracy was pretty good for a few of the places where we know we have hot spots. Okay, I'm going to go back. The places where we know we have hot spots, Humboldt being one of those new hot spots. This is where that 100,000 nanogram per liter sample I was telling you came from. So this is a new emerging place off Trinidad Headline. We slightly underestimated the DA values there. And then as you go further south, we do better in some places. Monterey Bay, we did pretty well. DA was overestimated in the Bight, where that bloom didn't happen. The Southern California Bight was one place where we did predict there wasn't much of a bloom, but we a little bit overestimated the domoic acid. And I should point out, and you will see this in a minute, that the model does not rely on having Pseudo Nitzschia in the water in order to have domoic acid. Because sometimes we don't see Pseudo Nitzschia in the water when we see domoic acid. So does the model work everywhere all the time? No, it does not. Just read our papers. Does it build off everything we know about Pseudo-Nitzschia and DA? Absolutely not. And I guess that's one of the points here, is that we've been trying to build something off of fairly scant knowledge, and with the hopes that it would work and be a good tool for managers. So Rick likes to say this a lot, and I think it holds a lot of punch. Rick Stumpf, sorry for stealing this, but it's so true. All models are wrong, some are useful. And George Box said this in '76. It holds a lot of resonance for me, just because when I think about modeling and we get frustrated that we're not doing well all the time, we have to remember these are tools. They help us evaluate our assumptions and our hypotheses. And I think that's what kind of keeps us going when it comes to the modeling and why we do this. We want to think about a framework. We know we're not going to get everything right in the ocean all the time, but we want to put something into a framework that allows us to test these questions. So when Stephanie Moore and Shelley Tomlinson and some others, we put together a review paper of HABs a couple years ago, and we basically stole Porter Hoagland's framework and kind of modified it a little bit, but we like this because of the emphasis on the human dimension. You've got the human in there. These feedbacks do clearly show how HABs and humans are connected, and that's sort of baked into the pie. But what might not be clear here when you see this in the paper is that this has a legacy. This comes from a way of thinking about the Earth system that might not be entirely obvious. And this goes back to the Brotherton diagram, which came out of a NASA study, 1988, saying, we have an integrated Earth system and we need to think about it that way. And we need to think about these ecological interactions, these environmental interactions, and how the ecology fits into that, and as well as how the human activities fit into it. So this was probably one of the first times where someone was saying, maybe this piece is also important, and then we need to integrate all of it. It seems trivial now. I think that we've all been thinking about Earth system science and Earth system modeling framework for many years, but I think at the time it wasn't taken for granted, and it spawned a lot of research over the last 20, 30 years. I mean, really, this report is what spawned JGOFS, GLOBEC, SOLAS, IMBER, all the big programs that we can think of that have taught us so much about the ocean. Microbial loop, that the ocean is a major sink for CO2. These would not probably have been national priorities if it weren't for this report sort of staging how we should go about and make the scenarios that would allow us to do those studies. But of course, the human activities are kind of marginalized. It's a little bit asymmetric, as you can see in this image. The coupled human-natural systems that we talk about now were probably not as emphasized. So as we go through this talk, and I will not be discussing the human dimension so much in my talk, but I do just want to say that this framework is going to kind of stage some of my conclusions, so think about it as we go through some of the modeling that's out there. And there's a lot of models. And so in that same paper, we went through and sort of said, okay, remote sensing detection algorithms aside, we're not talking about those right now because we just, it was already a 50-page paper. We'd need another 50 to talk about remote sensing. But there are a lot of models for HABs and there's so many ways to skin a cat. You could bring in a number of circulation models, a number of statistical approaches. I'm just going to go through these. You can see this was a very long table. And this doesn't even get into the models that just look at chlorophyll. This really was focused on the models that try to get at a certain species distribution, species population response at the HAB level for species. So I'm going to start with a simple approach, and this is what we've done in California. And this builds off of some of the earlier work on Pseudo-Nitzschia modeling that started really with the Bloom paper. This was embedded in a book on the ecology of harmful algae, where they took mostly lab experiments and matched up what the Pseudo-Nitzschia and domoic acid looked like with respect to all these environmental conditions, a little bit in the field, and then made some statistical models from that. And what you can see there is, in the green part of the diagram, are all of the parameters that fell out as important. These were generally negative relationships. I think most of the nutrients were negative relationships. Then we did the same thing in Santa Barbara. We had a long running time series of hydrography from the Plumes and Blooms cruise coupled with remote sensing, or I should say optics, because we have a radiometer that we put in the water for that. This is really a remote sensing project meant to kind of biooptically assess our measurements of chlorophyll with respect to things like MODIS and CWIFs. That was the origin of it, hence the sort of emphasis on the absorption at these various wavelengths that you see in the red part. But we also saw an association with salinity, chlorophyll, again, nutrients, both the Si to N and Si to P ratio. A student of Rafe's put together a similar model just for Pseudo, not for domoic acid, in the Monterey Bay, and saw some similar relationships. You're seeing the overlap of things that fell out as important here, which is temperature and nutrients. We didn't look in California at river discharge, but that was important in Monterey. And then other studies have not looked at the optics. So that has made our project a little bit unique, that we have this optical component. If you look at what is the best models that come out of this, if you're looking at your full suite of variables, the nutrients are there each time, that's why they're circled, as well as some of these optical parameters. But it's really hard to acquire these nutrient data in real time or remotely. We can't use these models for routine forecasting because we can't get at nutrients in real time. So we thought, well, let's be more heuristic about this and just decide on some optical parameters that fall out of these models that probably co-vary with all these ecophysiological important things that are happening, but they're going to give us this kind of a snapshot, and maybe we'll be okay at this. Maybe this will work. And again, I think this was our, when Al and I went through this talk, we did not, really fast this morning, I didn't realize how many things had gotten shifted in the PC conversion. So the idea here was that maybe we could attempt an Earth system modeling framework, but we were going to do it, we were going to cobble this thing together. Jim McWilliams was the best compliment I ever got from Jim McWilliams was that I'd cobbled this together. It is very cobbled. What you're seeing here is you've got circulation models matched with satellite ocean color imagery. You mix that all up in a bucket, you compute your HAB statistical model at those grid points, and then you can create a risk map. So in this case, we're using Yi Chow's quasi-operational ROMs for California. It's a 3-kilometer model. Satellite ocean color, which is MODIS, we're on Aqua, we've switched to VIIRS on SUMI and PP. And then the HAB risk map originally was developed just for Santa Barbara, so I've been using the schematic for a while. I haven't extended it out to California, but you'll see the California maps in just a minute. But first I want to just say that we had to reduce these models down, get rid of the nutrients, create new models that only considered things like temperature, salinity, and the optics. And so this was where I wanted to just point out Jamie Smith's poster because while she's already presented it, she said she'd be happy to take you back. Temperature over here fell out as negative for cellular domoic acid. She sees a nice, I would say it's a pretty strong relationship, a negative relationship with temperature in her 10-year time series from Southern California Bight, which we might not have expected, but considering that we have seen a trend, at least in Santa Barbara Channel, where this model was developed initially with temperature over time, and that really is that negative association, i.e. that as these waters are upwelled with high nutrients, that's when we start to see these blooms and even the DA production. All right. So one of the things that you have to contend with the satellite imagery is that you've got clouds. Daily imagery is tricky. We're trying to do this on a routine basis using daily imagery, so we had to contend with that. We weren't going to do 8-day means, so we decided to use this off-the-shelf program called DyneOF. This is Data Interpolating Empirical Orthogonal Functions. So it takes, similar kind of singular value decomposition that you would do for a regular EOF, but now what you're doing is saying, here are my dominant modes of variability. Can I use those to back calculate and fill in the missing pixels from this daily imagery? So we apply that. This little animation is just, this is actually one I think you need to click on, Al. I didn't point that out when we went through it, but that animation on the left is just to illustrate how in 2009, when we were initially starting this, we started to play around with the risk maps themselves and do some of that filling and then match those data sets up with our time series from the Santa Cruz Wharf, which did pretty well. So that's a very short time series. But we went into, goodness gracious, we went into full-scale sort of routine operations with this in about 2014. So what you're seeing here, if you could read it, Crossing the Valley of Death with the Sea Harm model, which is really reference to when you're going from research to operations and you're trying to sort of span that, I would say we're still crossing that valley right now. This was the first stage where you say, can we fit all the components together? Can we demonstrate that this works? So we had to go from the imagery to the DynEOF filling on a daily basis, get those error estimates, do some tuning of the chlorophyll algorithm that we have, I think, kind of abandoned because the regular off-the-shelf chlorophyll algorithm from NASA is working pretty well for these purposes. Then we do these nowcasts based on the data that we get from the ROMs and the satellite imagery. In order to create a forecast, we've tried different approaches, but the thing that's been really nice and computationally inexpensive is to take an advection scheme, a really simple advection scheme. We get the currents, we get the 3-day forecasted currents from ROMs, and we can just push those fields forward 3 days to get a 3-day forecast of the bloom. And that's been our focus, putting that out daily. And now we have to integrate this and really create a tool akin to a lot of the bulletins that you've seen already at this meeting, like for the Gulf of Mexico or for Florida, and then for the Pacific Northwest. And we're doing this slide doesn't call out all the different agencies that we're doing this with, but this is a very tight collaboration with NCOS. So Rick Stumpf is helping us develop that tool, and Yizhen Li is now helping to put some of these pieces together. We've been running this on high-performance computers in Wisconsin for a while in demonstration mode, and now we think we're going to have this live operationally at Coastwatch, which is a really interesting curveball for any of you guys who know stuff about operational modeling. That would be kind of the first time that something like that has happened for an operational model in the United States. So this is what it looks like on the website. We have these daily images of nowcasts. We have risk maps that alter the color bar scale just slightly to help people like fishermen who told us they didn't like the other maps or couldn't interpret them. And then there's a portal where you can pull down time series at any pixel that you want. And this has, I think, been really helpful for anyone who wants to do science or evaluate this model themselves. And that's run at Senkus. I'm at SCOOS. It's a little confusing, but we've had a long history running this at Senkus. We're thinking about seasonal forecasts, longer timescales, how to link this to crab toxicity, how to link this to shellfish or animals in the benthos. It's really tricky. We're really only getting a picture of what's happening in the water here, but it would be really nice to know how to link this to the food web. We're going to try to do it statistically. We've seen some nice association between some of our mean maps and what it looks like is happening in the water with respect to the water and the shellfish simultaneously. Here's a map for Trinidad and Monterey later, where you can see a nice matchup there too as that bloom started to shift north in 2016. And there's a lot of observation platforms that are required. We now have, Rafe has 2 IFCBs, one at Catalina Sea Ranch, the first offshore aquaculture site in the United States in federal waters, and then Monterey Bay, and he's also got one in San Francisco. But these are the 2 we're focusing on, along with in-water measurements and shellfish toxin measurements, to try to get all of these coincident measurements together and maybe put a statistical layer on top of our model that says, what's the probability of shellfish getting toxic? And this might not tell us anything about crabs. I think that's going to be trickier. As far as Monterey Bay goes, we have a model there. Rafe and I have been working with a grad student, Britt Stacy. Al, can you please play that animation? This is just a particle tracking experiment. Oops. So you need to hover the cursor over the little, if you go back up, over the blue Humboldt Bay region at the top. No? It's going. This is just preliminary work, early days, trying to understand why it is that we see such high concentrations of domoic acid off the coast there, off the Trinidad headline, but we rarely see the oysters and the mussels getting toxic in Humboldt Bay. It's one of the biggest oyster producers in the north part of the Humboldt Bay. So the first thing to do was to just see if physically those particles can even get there. After that, we're going to try to understand some of the biology and see what it is that prevents the shellfish from getting toxic. Because even if it's not a problem now, we do want to be able to say in the future, if let's say circulation or something does change, will we be able to get domoic acid into those shellfish in the North Bay? As far as the crab story goes, this has been particularly challenging because the fisher community is looking to us to say, well, you're measuring all this stuff all the time, you've got all these maps, can you please just tell us when the crabs are going to be toxic? You know, the 2015 bloom was extremely traumatic, and now this is on everybody's radar, and we have not been able to say, Do we know it's in the sediments? Do we know what those pools look like? How long will these animals keep getting recontaminated by domoic acid? We've tried to look at the maps and make at least some seasonal estimates, and we did have the Pacific Fishermen's Association call and say, what is your best guess? And at the time, I guessed right that particular day. I said, well, I think the first thing we're going to think about is Santa Barbara Channel and the Santa Barbara Channel Islands, because we saw the longest, most prolonged event there this summer. Highest toxins of the year were in Santa Barbara. And then sure enough, Anacapa Island and Santa Cruz Island is where we now have a lobster closure. I couldn't have told you it was going to be lobster, but I think that based on the work we had done, we had a good sense that this would be a hot spot. But there are lots of other places where we could see a similar thing, and they just have not manifested this year. But we do know that there's rapid downward transport of DA. It gets into those sediment traps, like I told you earlier. It gets into the sediments. It can persist for a long time in the sediments. So I think that it's going to be a challenge for this community to, I don't think we can just use statistics. We are going to need some measurements moving forward to make this connection to the benthos. It's going to be a big challenge. Skill assessment, the ultimate challenge for us with this model is really saying, how well are we doing? We have only pier stations to go off of, but what we've done, and this is just for the Santa Cruz Wharf, is to try to do those matchups as well as we can with a nearshore pixel and say, how well are we doing with that time series? Can we create a tool that maybe managers could use where they could shift around the number of what is 50% an interesting number, or should I care when the model says 70% probability? So we've tried to move that around where you could, optimize your accuracy relative to your false positive rate or something like that, depending on how you set that threshold. And we're doing this with the Senkus and Skus supported pier stations along the coast. There aren't really that many of those, and they are really just these bucket samples off of piers, so I think it makes it tricky to compare to a 3-kilometer model. I want you to go to Kendra Hayashi's poster, though, because she does take the model that I referred to earlier, that was Jenny Lane's model for the Monterey Bay, which is kind of a local model, and then compares that to the sea harm model that we're running for the entire California coast, and then compares that to the measurements from Santa Cruz Wharf and also from Monterey Bay. And I think that that's a nice illustration of some of the skill assessment and the difficulty that we've had with skill assessment. So this gets me a little bit then into how we're setting up our observing systems. Are we doing this optimally? Sergei Frolov wrote a really nice paper challenging this question using satellite imagery, a lot of satellite imagery, MODIS fluorescence line height from 2002 to 2008. And asked this question of, well, first we have to think about decorrelation scales if we're going to even think about how we're applying things like satellite imagery to our assessments of HABs along the West Coast. And he had to examine what that decorrelation scale looks like latitudinally. What does that really mean? Well, that's really just in math terms, that's just the zero crossing of the autocorrelation function. But I guess in more plain language, what we're saying is when are these pixels no longer related? When are they no longer autocorrelated within a certain region? And so you can look at the gaps in the MODIS imagery and evaluate the distance, the autocorrelation between all those pixels. And in this case, he said the smallest was off Central California in Santa Barbara Channel. So that's sort of where you have your best connection. But interestingly, places like the Southern California Bight actually do have a lot more fog than we might have thought. And cloud cover is actually quite extreme, and it was harder to make that connection there. But the end result is he looked at the temporal decorrelation scales, the spatial decorrelation scales, and said if you make a synoptic R-squared, what does that tell you? Well, what he found was that the duration of those bloom events decreases from south to north. So you got shorter blooms as you go north, and so that means how useful the imagery is also decreases south to north as a result of kind of combining all those statistics. And so this was interesting when we started to think, too. He was able to take all that information, combine it with some more statistics, and then say, are we optimally sampling for HABs to capture all the variability that's in the system? And the answer is no. If you just look at the existing stations, particularly those triangles, which are our Senkus and Skus, the IUS-funded pier stations, we were only capturing 20% of the variability. And that would be a good estimate now, because we've actually had to decommission a lot of these sites in recent years because of lack of funds. So then the circles are the proposed stations, which were generally offshore, average 15 kilometers offshore, give you maybe more like 60% of the variability in the blooms if you use those. And those would be great for us, because now we would get to a place where we could truly see, not only tune the models better, but understand if we are really getting this kind of ecosystem level variability correct with the model. And so you're seeing the existing network there on the right, and these are the proposed stations. Would this be perfect? No. We still need monitoring. We're still, I think, this is not what Congress wants to hear, but we need all of this. We need all of these stations. We need the offshore stations. We need the IFCBs and the ESPs. And then we need the state also, of course, sampling the shellfish routinely to make sure that we are, safe. So this is the challenge. This is the message that we have to, I think, put forward, that observing is important. And this is why I love the Pacific Northwest HAB Bulletin, because it does really nicely pull together everything that is available. You have all of the in situ work, and so this includes a lot of the beach sampling from ORHAB, and then the ESP. Gosh, everything got jumbled. I'm so sad. At least we can see you, Stephanie. The ESP moored off La Push, and this is work that Stephanie's been leading, and this is a partnership with Nanus trying to transfer that technology to Nanus in the Pacific Northwest. And then they also take some of the work we described from the McCabe paper and thinking about long-term forecasts. What do things like temperature mean for those longer-term forecasts? Potentially there's a threshold right around Oregon that makes this very important. And so you get kind of a short-term risk, a long-term forecast, and this is not an Earth system model, and nor does it need to be, but all the components are there if we wanted to create that framework later down the line. You have a really nice circulation model that Parker McCready put together, Live Ocean. This is coupled with a biogeochemical model by Neil Banas called the Cascadia model. So you've got your coupled physics and biology, and this has been really useful for predicting these blooms, because a big part of this, as I said, was that onshore flow. So bringing DA to the coast, can you even see that? Is dependent on various retention and onshore flow mechanisms. So this is another little animation that I thought would just go automatically, Al, but it didn't. Hello, hello, hello. So this was something that Vera supplied to me. You would be seeing an animation of these particles moving down the coast, and this was done in advance of the big razor clam dig last weekend, right, Vera? What was the end result there?
We have closures in Oregon now, so those particles in the south made their way to the coast.
They did. So, so just trust me. This would have been amazing. You would have seen all these particles coming down. It would have been verification, but here we are. Another place that I've worked is the Chesapeake Bay. There are a lot of models, a lot of opportunities to do Earth system forecasting or system modeling in the Chesapeake Bay. This project's no longer funded, but it's one that I was directly involved with, so I thought I would show it. It's called the Chesapeake Bay Forecasting Project. Used a ROMS configuration and nested ROMS to look at the physics. Was forecasting, this was also highly coupled with the atmosphere and also with a WRF model, so looking at watershed runoff. And we had 16-day forecasts of things like temperature and salinity. We also had it coupled to a pretty basic biogeochemical model, the Kasha-Fenno model, that gave us nitrate, phosphate, which turned out to be all we needed to, this is so depressing, all this jumbled, to predict Pseudo blooms. We did a very similar approach to what I showed you for California, pretty basic generalized linear model. Forced it with what was important, which in that case did tend to be more the nitrate and the phosphate in the Chesapeake Bay, as well as temperature and salinity. Heidke skill score is a pretty stringent way of doing skill assessment. The probability there was only 53%, but our probability of detection was pretty high at 75%, and we had the luxury of having this very long MDDNR time series to build this from. But we didn't have a lot of domoic acid, so even though there are some, I would say maybe even argue some chronic domoic acid exposure in the Chesapeake Bay, there really hasn't been any kind of a shellfish closure or an ASP event that we're aware of. But this could be used, let's say that changes, we do have the building blocks in place to create a forecast of Pseudo in Chesapeake Bay. So now I'm just going to go to the new kid on the block, the Gulf of Maine, where we have this emerging problem. Funds were dispensed by NOAA in 2012 to start looking into this. And then 2016 and 2017. Now we have two, I guess it was only the second recall of shellfish in Maine history, but there have been these two major closures. Correct me if I'm wrong, Don, but I think it was like something like 60,000 pounds of blue mussel that were recalled. So it's pretty extreme, and managers are reacting in an extreme and swift way and saying, we're going to create protocols, we're going to close things even at a hint of toxic Pseudo. Maybe if we get some models there, they won't have to be so draconian. And then Rhode Island also saw this. So either this bloom then went downstream, or these were concurrent blooms. But something also happened simultaneously in the fall of 2016, spring of 2017 again in Rhode Island. And we saw these shellfish closures all along the coast there. So the Gulf of Maine has been hit pretty hard. What do we know about Pseudo in the Gulf of Maine? Well, there's not really a lot out there in the literature. This is a nice paper that illustrates what species assemblages are there or were there when this work was done. And these are some nice surveys. You can see, I'm going to blow that table up a little bit so you can see that there are a few species that are capable of producing quite a bit of domoic acid in the Gulf of Maine. P. seriata was the worst, but then we've got all this cryptic diversity. So using omics, they were able to illustrate that there's another species, sort of unidentified Gulf of Maine species that is also pretty toxic. And so I'm not going to give the punchline here, but please wait for Kate Hubbard's talk, which is right after this. And she's going to tell us who it is that caused this last bloom and the bloom before that. So there are things potentially changing in the Gulf of Maine, and so these researchers are looking into this. This is led by a lot of different people, but Dave Townsend has a long history. That's JMS 2015, but you can't see it. He has a long history of examining, with his collaborators, the confluence of these different water masses. You have the Scotian shelf water coming down the coast. Near shore, this is cold, fresh, nutrient poor relative to the deeper slope waters coming up from the Gulf Stream, the warm shelf slope waters, and then the Labrador Sea slope waters that are coming in. And a lot of things are changing. There's definitely an influence of glacier melt on the Labrador Sea and slope waters, but then there's also seemingly a strengthening of these Scotian shelf waters, or at least a stronger influence. And you'll see that in some of the next slides. Looking at the Jordan Basin and then George's Bank, I will show you some of this work. This is recent. It's a 2014 paper, even though you can't see it. So looking at those stations, 114, 43, those are the two I just called out. A lot of surveys. This is so sad. This is really sad. A lot of surveys looking at these profiles, and what you're seeing are those relative nutrient contributions, particularly as those different water masses are mixing throughout the water column and those different contributions. It is a really complicated system. And you can see how the extent of those waters changes, how much they do or do not mix as you go down in the water column, and how that can influence what the ratio of those nutrients looks like, what is the temperature, how fresh or how saline is that water. And so some interesting observations have been made about what happens as those water masses mix. And I think we don't exactly know what this is going to mean for Pseudo. This will have to be ascertained, but Yuzhen Li and Roying He had a really nice paper looking at a long time series of MODIS imagery and saying, if we look at the temperature chlorophyll, we apply that DyneoF program that I mentioned earlier, fill in all of the gaps, which is definitely a capability that now exists in the Gulf of Maine, and then look at the dominant modes of variability that come out of that DyneoF analysis. Some of them really did seem to fall along the lines of at least were correlated nicely with things like the slope water and the Scotian shelf water. So you have these modes of variability of productivity of the biomass that does seem to be driven by, these various components. And so the question is, if you've got this sort of barotropic pressure gradient set up by the Scotian shelf water coming down that kind of can impede the deep, more nutrient-rich water from coming in, what does that mean for the blooms if these things are kind of oscillating on interannual timescales? Can we connect sort of these emerging things like Pseudo blooms to this competition of these different water masses? And if it does connect back to things like an increase in importance of the Scotian shelf water, I feel like you can kind of see this sort of interesting thing in the amplitude time series here of the EOF mode showing something like that. And I believe Yuzhen Li linked the slope water mode to the NAO. So if things are changing kind of decadal timescales in the Atlantic, we might see that manifest here too. So we don't know what all this means for Pseudo, but luckily we've got people on the street figuring it out. got the circulation models, nice remote sensing, observations, a great team, ESP, it's going to look at both Alexandrium and Pseudo, and maybe even get a chance to apply some off-the-shelf models, like I would argue maybe the California model could be applied in a very quick and dirty way to find out if it works in the Gulf of Maine. We do have some precedent for that model being applied to other places. A group of NASA students put this together for a summer project. This was in response to a southern right whale die-off in Argentina, and so they took the model, got as much imagery and data as they could, and applied it to that region, and did show a pretty nice correspondence with where those southern right whales were stranding in Argentina and how that matched up with the model. And they won an award for it, which was very sweet. And so I'm just going to bring you back in summary to this ESMF framework and how we can think about putting marine HABs and freshwater HABs into this. And we could, I think this has a lot of power, because outside of creating tools that I think are very important for managers, we can make quick and dirty tools, we should, but I do always want to have this in the back of my mind, which is that I want to make models that fit nicely into this framework so that we can do things like hindcast analyses, process studies, test our hypotheses in the absence of the kind of money that we need to do major process studies these days. These models could be very powerful. We can do powerful projections, to the best of our knowledge, using the ecology and the ecophysiology that we know now. And we can really bring in the coupled human natural systems in a new way. And so that, of course, I want to just make a plug there for Stephanie's talk today at 10:15. She'll be talking about some of the work she's doing on that kind of socioeconomic and human dimension side, and we can't ignore that. That has fueled huge programs today like Future Earth. And so when I look at this, I think, how can we do this? We've put a little bit of effort into this already. So in summary, I'll just point out, we have done some work. I presented this model some years ago at this HAB conference. We created a 0D model that would hopefully use some of the things we think we know about mechanism to do this in the absence of statistics. And so you're seeing our proposed model here. We were a bit thrown off, given a curveball by a model that came out simultaneously as we were working on this in 2013, that does things in a much more complex way. So then we thought, well, now that we have a model that we need to contend with, our model's much simpler. Can it be simpler and still get at the variability in these blooms and in domoic acid? We want to test this. We didn't end up publishing it. We did a lot of work comparing these two, showing how well they work relative to one another with respect to chemostat cultures and other things, but then we didn't do the coupling. We haven't been able to secure the funding, but there's been a lot of discussion about working with Chris Edwards' configuration of ROMs with a Nemero model. It's a moderate complexity ecosystem model. This is a 9-kilometer model, but it's a real-time model, and we could really start to ask some questions about whether we can capture DA variability with this 0D model that we have, if we can embed it. We've also proposed to work with more of a hindcast model to do some process study work with hindcast. This is a 78 to 2013 model, 2-kilometer configuration, but pretty nice resolution, really captures some mesoscale variability. And this is coupled to the biogeochemical elemental cycling model that Daniele Bianchi is now working on with Jim McWilliams. And I think that there's a lot of power in this, because if we can start to make this kind of an end-to-end system run in parallel with the things that we know are working pretty well for managers right now. But we can do this in parallel. We can start to ask some pretty big questions about how these blooms have changed over time. Will they change in the future? And I think that's harder to do with some of the statistical models that we're running in real time right now. All right. What does the future hold? There are good reasons to do this. If you look at the recent climate science special report that just came out with the CMIP 5, RCP 8.5, projections, they're pretty sobering, in my opinion. We do see quite a bit of temperature change, and we know this is co-varying with all kinds of things. The biogeochemistry is going to be changing too, and we need to start thinking about what does that mean for HABs. So this just fell into my lap as I was putting the talk together, so I thought I should show it because it cracked me up. And I was thinking about, I don't know if you can read the gray font, where HABs should fall, and then we can debate this. I would have said we could debate this at the banquet while dancing, but that's not happening. I don't know where HABs fit, but when I saw this, I thought, oh my God, my kids totally fit on. I'm pretty sure that my oldest is here on this axis. My youngest is definitely here on this axis. And so I don't know what the future holds for HABs, but I know I'm in for it. And so I want to end by saying these are the people who have influenced me a lot. The funding, the people. This all comes together on one slide. I've got a lot of people to thank for the way I think about science and why I'm doing what I'm doing with the modeling. And then, of course, the funders. This has been a recent development over the years. Well, actually, I shouldn't say that. Going back to the early years of even developing this model when I was doing my dissertation work, it was a NASA Earth Science Fellowship that supported that. So NASA has been in the picture for a long time, but so has NOAA. A lot of the R&D that goes into this, we couldn't do this without the ecology, and so NOAA has been a huge player in that and funding some of the early development of the model. And then we've had some state support, and then more recently, because I'm at SCOOS, and also we have the HabMap run mostly out of SCOOS, but with funding from SYNCUS as well, they are very important in keeping this going. And so I was going to end with this slide of us dancing in 2009. So Kathy's not here, so I can show this. And that was a great meeting. So I just want to say, as I close, because again, the Santa Cruz in me is going to get verklempt, but I wanted to just say that this meeting is so amazing for the way that it brings us together. And we have this moment to have these collegial relationships that are highly intellectual in nature, but also make really great friendships. And so I was going to end with the words of David Bowie, but I guess it's going to be a little more jazzy tonight, but it'll still be fun. So I love you guys, and thank you for the opportunity. And my apologies for the slides that were impossible to read.
Questions? Yes, we miss Rafe and Kathy.
Clarissa, thanks very much. Why do you think your model works less well moving from south to north?
So that wasn't the model. That was really just evaluating how well we can use satellite imagery as we move from south to north. And you've seen this, in your own work. You know that satellite imagery is tough given how ephemeral the events are, and then the fact that you have a lot of cloud cover. But I didn't show the work that you've done with Rafe applying DynaOF where there is success there.